Hello, I'm Kim Schreiner. I'm the founder and president of the board of the Phil Simon Clinic Tanzania project. I'm also an infectious disease specialist and have been dealing with the coronavirus pandemic at our hospital at Huntington Hospital. I was asked to do a presentation on the vaccines that are now available for SARS-CoV-2, and I'm delighted that the Phil Simon Clinic Tanzania project has asked me to host this lecture as it's an opportunity for us to highlight some of the things that we have been able to do for our project in spite of the pandemic. In the last year, we have recruited 5 new candidates for our scholarship program in Tanzania. That includes a physician for medical school, a nurse for nursing school, and 3 social workers. This has been thanks to the hard work of our scholarship committee. All of these individuals are in Tanzania, and they will be attending university over there to continue their adventures in healthcare and provide much needed care for their people. In addition, we've also proceeded forward with our plans to build the Kisongo Center for Health Sciences. This is all in the backdrop of not being able to travel and with the pandemic occurring around the world. So it's really pretty remarkable that the board and the incredible staff that we have both in Tanzania and here in the United States have continued to show their commitment and compassion and have been able to accomplish these really remarkable things in the last year. We understand that Tanzania is undergoing many of the same problems that we've had here with their own pandemic. And of course, as a very poor country, they are underserved in terms of their ability to test, isolate, treat, and most importantly, prevent this disease. So we hope that as we move forward for the next couple of years, that the vaccines will become available to much of the world that truly needs additional healthcare and help, and certainly to prevent the spread of the COVID-19 pandemic. With that, I'm going to go ahead and start our lecture on the SARS-CoV-2 vaccines. This lecture was originally given on January 18th, 2021 to a live audience. I will try to recreate some of the questions that were asked during that period after the lecture to address the issues that are on people's minds. We know that plagues and pestilences have been around since the beginning of humanity. Thucydides talked about different prehistoric plagues, and especially the plague of Athens, which was likely a typhoid plague. The physician Galen described the smallpox plague that hit Rome and actually caused the downfall of Rome with the death of Marcus Aurelius. Certainly the Black Death that circulated through Europe much in the 1350s and 1400s, actually all the way up into the 1600s, probably caused mortality of 25 to 50% of Europe, and is a very important pandemic in the emergence of from the Middle Ages to the Renaissance. There have been pandemics and plagues in the Americas as well, largely brought by the European settlers. Smallpox, of course, has continued, was one of the most significant events, as were measles and other hemorrhagic viruses. So this is not anything new to humanity. We have had many modern pandemics as well. Of course, the biggest one was the H1N1 Spanish flu pandemic that ravaged the entire world, infected over 500 million people, and caused the death of 50 million people between 1918 and 1919. It was a similar pandemic to what we're experiencing right now with SARS-CoV-2, in that it tended to have surges, the worst of which was in the fall of 1918, and caused a high degree of mortality as the troops were returning from World War I. There have been smaller pandemics through the last century, not the least of which was the HIV pandemic, which of course is still ongoing, that started in 1981. We now have terrific treatment for HIV and can control the pandemic that way, but we still don't have a vaccine. On December 31st in 2019, China alerted the World Health Organization about several cases of an unusual pneumonia. And in January of last year, 2020, remember it was only just last year, it seems like a century, a previously healthy 54-year-old woman died of a mysterious pneumonia in San Jose, California. And so it began, and we all know how this is played out. The cause was SARS-CoV-2, which was a new novel beta coronavirus. It is the virus that causes the disease COVID-19. Prior to December of 2019, no one on the planet had any immunity. It has been the cause of the COVID pandemic. It is a highly transmissible and very infectious pathogen, easy to transmit from human to human. It is very dangerous and caused millions to become sick and some to die. There are limited medications to treat it, and it is certainly not a hoax. And as you can see from this remarkable animation provided by the New York Times, it's a small viral particle with these spikes that occur on the outside of the virus, the so-called spike proteins, which have now become a focus for the development of a vaccine. So we all have become very familiar with this terminology to describe what is a brand new pathogen. We know that this was a zoonotic spillover event. Beta coronaviruses are largely in animals. They don't usually infect humans unless there's an event where there's a zoonotic spillover. This did happen before in 2003 with the first SARS infection. It also happened with MERS, Middle Eastern respiratory virus, which also is a beta coronavirus transmitted likely from bats into camels and then into humans. SARS and MERS have thankfully not been terribly common infections, though they have a very high mortality. But this event probably happened sometime in China. Whether it was at the Wuhan wet market or not, we don't know. It may well have been just a person who encountered a bat that was infected with this virus. It jumped species. When viruses jump species, they often become nastier, more virulent. Often they also have an intermediate host. In this case, it's likely the pangolin, which is an interesting armadillo-like animal who is a high-value item in exotic animal markets, but also can serve as a reservoir for viruses such as SARS-CoV2. Again, coronaviruses commonly occur in bats and birds. They can be transmitted to other mammals, swine, cats, dogs, camels, and then cause disease. This particular coronavirus is in bats, transmitted into perhaps pangolins and then to humans, and we know the rest of the story. We're still living the consequences of that event, and it's been largely a very significant factor in around the world. No country is spared. We even now have cases of COVID-19 on Antarctica. It was sort of the holdout continent for a while, but it too has now been, we've had reported cases at some research stations there. So we're very familiar with these images. This is an image that I often see every day when I'm in the hospital. It's really been a terrible catastrophe, a humanitarian crisis, and the most significant pandemic really for the last 100 years in terms of its effect on the global community. These are the statistics as of January 17th, really pretty horrific. Over 95 million cases of confirmed SARS-CoV-2 infection, COVID-19. We know that that's probably an underestimate because there's probably another 20–30% of people who die from unknown reasons that actually are related to infection with SARS-CoV-2. There have been over 2 million deaths throughout the world. The United States has been heavily impacted by this pandemic with 24 million cases. And as of today, January 19th, 2021, we unfortunately passed the really horrific landmark of over 400,000 deaths in our own country. California has been impacted by this pandemic to a large extent, especially recently, with over 3 million cases and 33,000 deaths. LA County, of course, is also a very high density area for this disease with 1 million infections and about 13,000 deaths, and those numbers continue to rise, which is really daunting. There perhaps is a little bit of good news in the last couple of days in that we are beginning to see that we might be going over the crest of this tsunami of disease with a slight decrease in the number of percent positive rate in the last 7 days. So that may be a good sign that we're through, or at least beginning to show some signs of improvement in the horrific numbers that we're seeing here in Los Angeles County and throughout California. It's very likely that we will continue to have frequent surges with this disease, especially when people gather together, either moving indoors during cold weather or gathering together for holidays, which we of course have tried to discourage, but human nature is human nature. It's a hard thing to change. And so we anticipate to continue to see surges really around the world for the next several years. That's the nature of these type of pandemics. So we're going to have to do something to control this pandemic besides just let it rage on. During this time, we have learned a remarkable amount of information about this disease. We initially thought it was only a droplet-borne pathogen. It's turned out that it is actually an airborne pathogen, highly infectious, can be transmitted very easily from one person to another simply by talking or singing or yelling. It is a very aggressive virus. These are the spike proteins that are on the surface of the virus. They are the attachment site of the virus to the human cell, the ACE2 receptor, which is widely expressed in your nasopharyngeal area, your upper respiratory tract, and then of course, we know that it can cause terrible disease. It does certainly affect the lungs, but that's not the only part of the body that can be affected by SARS-CoV-2. It has significant cardiac effects. It can affect blood vessels and cause clotting, which can lead to stroke and heart attack. We know that it affects the brain. It can produce confusion and seizures and inflammation of the brain that can persist even after people have recovered. Many of my patients describe a persistent fogginess in their brain many months after they've recovered from the infection. The liver and kidneys are not spared either, nor are the intestines, and so it really is a very significant pathogen that has a great deal of effect on the immune system. As is true of all infectious diseases and all pandemics, the poor and lower disenfranchised parts of society are affected disproportionately. This is certainly true with the COVID pandemic in the United States, where it has highly affected lower socioeconomic communities and also people of color. And why is that? Well, it's certainly because of perhaps decreased access to healthcare. Many people in these communities are essential workers. They have jobs that they have to go to. They can't afford to stay home. They may live in multi-generational homes where transmission is easy for it to occur. They may not have access to information about how to prevent the disease or to be tested. And so there are many complicated factors that contribute to the inequity that we're seeing with this pandemic. And it certainly brings to the surface other inequities in society, as we have certainly seen in our own country over the last year. We do know how to control this pandemic fairly easily with inexpensive methodologies that work very well. We know that masking people certainly protects people from being infected with the virus and from infecting others. It both affects transmission and reception, if you will, of an infected virus. The masks are important and they certainly are a very inexpensive way of controlling this disease. We know that practicing social distancing, staying 6 feet, perhaps 10 feet away from individuals who might be infected is prudent, avoiding crowds and congregate living situations, and washing your hands. All very simple things, all things that are highly effective in preventing the disease. We also know that it's important that public health departments have the finances, resource, and infrastructure to do contact tracing. That has been a problem here in the United States. We haven't spent a lot of money on public health prior to this pandemic, and they have been really hard pressed to try to handle all of the contact tracing and overwhelming amounts of disease that have been going on. I really think that the public health individuals have been some of the most courageous and important parts of trying to control this pandemic, but they have certainly at times kind of come under tremendous criticism when their only interest is to try to prevent the disease from happening. We did find our way out, I think. This is what this talk is about, and that is the emergence of 2 very effective and perhaps 3 or 4 very effective vaccines in the last few months. There are certainly many vaccines on the market, and it is very clear now that vaccination is probably our only way out of this pandemic, and so that's what we're going to focus on for the rest of the lecture. On January 10th, very early during the pandemic, the Chinese government released the genomic sequence for SARS-CoV-2. They posted it on the internet, available to anybody who was interested, which was an extremely important move to try to generate intellectual curiosity and scientific development for possibly for vaccines and or medications. It's actually a relatively simple virus. It has 30,000 amino acids, and it was noticed very quickly which ones control the production of the spike protein, and that became a very important bit of information for scientists in terms of designing the vaccine. On January 11th, the day after the vaccine, the genome was produced by the Chinese, Dr. Barney Graham, who is a distinguished vaccinologist at the National Institutes of Health and a good friend of Dr. Anthony Fauci. He immediately began designing a modified protein that could be used for a vaccine. He saw the importance of the genomic sequence and how it could be manipulated to make proteins that could eventually be inserted to use for a vaccine. Dr. Graham was also aware of a small company named Moderna that was interested in creating vaccines for using a messenger RNA platform. Messenger RNA technology has been around for a long time. I will speak about this in just a few minutes, but it had never been used for a functional vaccine during a pandemic. Other vaccines have been tried with mRNA technology and came to fruition, but the pandemics that they were targeted against, specifically Zika and to some lesser degree respiratory syncytial virus, were not large pandemics, and so there wasn't an opportunity to test them. But now we have that opportunity. So what is a vaccine? A vaccine is an injection or substance that when introduced into your body stimulates your immune system to create protective molecules, which we call neutralizing antibodies, or cells that prevent you from acquiring or getting sick from an infectious disease. What do vaccines do? Well, they protect an individual from an infectious disease. They may protect a community from the spread of an infectious disease, which are probably the two most important factors with regard to this pandemic. And they also occasionally can be used to treat an infectious disease. There has been the development of a reasonably good vaccine for Ebola to not only prevent infection with Ebola, but also to treat those individuals who might be infected. The theory about vaccination has been around for thousands of years. It certainly was recognized in ancient Egypt and in the ancient Chinese culture that sometimes taking a small collection of infected material and making it into a powder and blowing that into a person's face might somehow protect them from developing that disease. They didn't know why that occurred, but that basically was an early form of vaccination, or what was then called variolation. Edward Jenner was a British physician who is accredited really with sort of establishing the first vaccine in the 1700s. Smallpox has always been an important pandemic virus, and it was certainly has raged through Europe over many, many centuries, but certainly during the 18th century when Jenner was alive. And he noticed, however, that milkmaids did not seem to come down with smallpox. And he noticed also that milkmaids often had some lesions on their hand. And those lesions came from sores that were on the udders of milk cows. A different kind of virus called cowpox, but very similar to smallpox. And so Jenner hypothesized that maybe they were getting small amounts of the cowpox virus, which prevented them then from getting smallpox. And so he did an experiment where he took a small bit of crusty material from a cowpox lesion, and he volunteered the son of his gardener, 8-year-old James Phipps, and he scratched a little thing into his arm and injected the cowpox scrapings. And James Phipps never developed smallpox. And that was the first really effective vaccine. And to that date, that's how we do a lot of smallpox vaccinations. Many of us have a scar from the smallpox vaccine that was given when we were children. So the requirements for a successful vaccine is that it be safe, that it be well studied, that it be directed specifically for the pathogen, whether it's a virus or a bacteria, especially the most important part of that pathogen. In this case, it's the spike protein of SARS-CoV2, that it produces a long-lasting and very strong immunity in all age groups and all risk groups. That includes both parts of the humoral or antibody part of the immune system, as well as the cellular immune system. That it be well accepted by the public. There are many people who are fearful of vaccines or who have religious or other reasons for not wanting vaccines, and that may be a challenge in terms of trying to achieve a widespread vaccination program. It needs to be able to be produced in large quantities. It should be easily stored and transportable, and it should have limited side effects. How do you administer a vaccine? Most of us are familiar with the intramuscular way of doing that. We all get shots. These vaccines are intramuscular, but you can also give vaccines subcutaneously under the skin. There are some that are inhaled. There's an inhaled influenza vaccine, for example. There's oral vaccines. We all recall, perhaps those of us that are old enough, the sugar cube with the polio vaccine, but there's also an oral typhoid vaccine now. And then there's some ingenuity being developed. Novavax, which is actually also marketing or soon to market a SARS-CoV2 vaccine, has produced this microsystem where there's little needles, it's a little patch that you put on your arm, and it injects small amounts of the vaccine over a longer period of time. So what do you hope to see? Well, we hope that the vaccine is well tolerated, that it has minimal serious adverse events, and that it produces less disease in the population that's vaccinated. We want to see, again, a robust molecular and cellular immune response that's protective against the disease, or at least diminishes the disease if you get infected, even if you've been vaccinated. Well, how do we know that this concept will work? So I'm not a big advocate for animal experimentation. We have a long and very sad history of abusing animals in this context. And there are really most humanitarians and animal advocates don't support ongoing animal experimentation. That being said, I think even some of those individuals recognize that occasionally there is an emergency that is of such magnitude that it may warrant a small, well-controlled, and very compassionate study of animals for the development of this particular vaccine. SARS-CoV2 does infect other animals, but they often don't get sick. And in fact, rhesus macaque monkeys, which are the animals that were used in this experiment, don't really get that sick. They don't necessarily die from SARS-CoV2, but they do get a disease similar to what humans do. And so this study was done earlier in the year, where a DNA vaccine, which is similar to the mRNA vaccines, was developed. 35 rhesus macaque monkeys were recruited for the study. Half of them received the vaccine, half of them received a sham or placebo. And then this vaccine was designed targeting the spike protein, just like all the vaccines that are being in development now. And then the experimenters looked at the humoral and cellular immune response of these monkeys in comparing the ones that had received the real vaccine versus the ones that had received the sham. 6 weeks after vaccination, all of the animals were exposed to large doses of SARS-CoV-2, injected actually right into their nasopharyngeal areas. And then the scientists looked at their viral and cellular response in the study animals. It was shown very solidly that those animals that had been vaccinated with the DNA spike protein against SARS-CoV-2 were able to control the infection and did not get sick. Those animals that received the sham vaccine got sick. But the duration of the protection is not clear, and that still is a question for humans. These are the data from the monkey trials, and I only show them to you because one of the big questions will be, do the vaccines prevent the spread of the disease? We know the vaccines can protect you from getting these symptoms of the disease, but you still may be able to spread the disease by carrying the virus around in your nose. And this is some early data that might be helpful down the road to show that these monkeys were able to control the amount of virus in their nose, even when they were injected with the material, by mobilizing local immune responses. So that may be important going forward with our own vaccine analysis. Again, I would always recommend that animal studies should only be done if the value of the study is going to clearly save millions and millions and millions of human lives and or animal lives. And that's when it's valuable. In the interim, animals should be kept in very humane conditions, and there should be strict control over the supervision of those experiments. But I do think in this situation, this may have been an exception to that philosophy. So the vaccine that have been developed now undergo what are called phase 1, 2, and 3 trials, and I want to just go over those so you understand what that means. A phase 1 trial usually is comprised of about 100 people or so, and it monitors the safety of multiple doses of whatever your medication or vaccine you're looking at. Phase 2 trial is usually a larger number of people, about 1,000 people, and that's a fairly effective way of looking at whether the vaccine or medication is going to have some effect on the population. And then you move to a phase 3 trial, which is the largest trial. In the case of Moderna and Pfizer, these were very large trials, about 40,000 patients in each trial. I will tell you that people who volunteer for studies are often not necessarily representative of the general public because they tend to be rather obedient people who are interested in participating in something like this. So you already have a little bit of bias. But both Moderna and Pfizer, and also AstraZeneca and Johnson & Johnson, have tried to recruit individuals that reflect the general population in age, diversity, gender, and comorbidities, the presence of things like diabetes or high blood pressure, which might be important morbidity factors with COVID. So how were these trials done? Well, basically we had half of the group received a vaccine, half of the group, and half of the group received placebos. And this was done in the context of a large amount of circulating disease. These studies were started in the summer. If you recall, we did have some rather large surges, especially in the southeastern United States during the summer. And so there was plenty of disease that these individuals are going to be encountering. The vaccine trials are monitored by an independent review board. These are usually a small group of scientists, maybe around 10 individuals, are the only ones who have access to know who's got the vaccine and who has the placebo. That information is not shared with the pharmaceutical companies, nor do the pharmaceutical companies have any power over the monitoring of the study. When they have achieved a certain number of cases of COVID-19, in this case about 200 cases, then they unmask the data and they look at, okay, of those 200 cases, who had the vaccine and who had the placebo. And what's truly remarkable is that both the Moderna and the Pfizer vaccine trials had incredibly similar results. They had about 197 people who acquired COVID in both of those groups. And in each one, they had only 4 or 5 individuals of those 197 who had received the vaccine. So that tells us that the vaccine was highly protective at a rate of about 94% for Moderna, 95% for Pfizer. That was way more than anybody expected. Everybody was very thrilled with that result, because that 60 or 70% certainly would have been acceptable, and is what we kind of achieved with the influenza vaccine. We want to make sure, again, that when these studies are done, that there is transparency with the Internal Review Board, which is the board that supervises the conduct of a human trial, and that there is leadership throughout the government and transparency, so that we know these studies are done well and done safely, and that they are reflective of the entire population. We also want to use these studies going forward, keeping track of the data, to be vigilant about what might be long-lasting side effects. So as I mentioned, most of the, if not all of the vaccines right now for COVID are targeted to the spike protein, which is on the outside of the SARS-CoV-2 virus, the most important part of the virus, allowing its attachment to the host, the human ACE2 receptor cell. These are the different types of vaccines that are currently being studied, and I won't go into too much detail except for the 2 that we're going to look at, which are the nucleotide-based vaccines, the mRNA vaccines, and just a brief bit about viral-based vaccines. This is a more common modality for vaccine development. That is the AstraZeneca and the Johnson & Johnson vaccine. These are utilizing a sort of a attenuated or less strong virus like adenovirus or pox virus, and that's the vehicle to transport the information about the virus that you're making the vaccine into the body. It's a very conventional way of doing vaccines. It's been well studied for a long time. It's pretty effective in developing vaccines, although we've had some failures, especially with HIV. But it is a tried and true methodology. It does have its own problems, of course. The nucleotide-based vaccines, as I mentioned, are relatively new in terms of using them as vaccines, but the technology has been around for quite a while. For those of you interested in following coronavirus vaccines, the New York Times Vaccine Tracker is a great way of looking at that. You can see that there are many vaccines that are being looked at, some of which are in various stages of study and development. There really are only 2 approved in the United States as of this moment. Those are under what we call EUA, or expanded or emergency use authorization. It's not an official FDA approved stamp, but it's that during this situation we think that the data supports the use of these medications because it will save lives, and indeed that's likely the case. Again, this is another way of looking. There's about 5 different ways of making vaccines for all different kinds of pathogens, including SARS-CoV-2. Right now we're going to look at the DNA and RNA ones. These are the viral vector ones. That includes the AstraZeneca vaccine and the Johnson & Johnson vaccine. These vaccines are still being used quite a bit. The Ebola vaccine that I mentioned that can be used for both prevention and treatment is an adenovirus-based vaccine. Excuse me, I think it's a canary pox-based vaccine. At any rate, it's a viral vector vaccine. But that's a common platform and will be some utility. These vaccines tend to be not as delicate as the mRNA vaccines. They don't require the same robust storage techniques. And so they may well be a better choice for countries that don't have good refrigeration. But we're going to focus today on the DNA and mRNA vaccines. What's interesting is that many of the vaccines that are being developed for SARS-CoV-2 have had very important histories with women scientists. And the 2 particular ones we're looking at here the Pfizer vaccine, Dr. Katalin Kariko is a Hungarian scientist. She actually is one of the pioneers for mRNA technology. She recognized more than 20 years ago the value of this technology and was able to overcome the biggest hurdle at the time, which was how do you get that little piece of mRNA into the cell so that you can give that information to the cell to make the protein that you want it to make. And she recognized that it had to be in a special little molecule, the little fatty molecule that we'll talk about, that carries the information in. So that was really pivotal work, and she was the main purveyor of that. She now works with Pfizer. Dr. Kizzmekia Corbett works with Moderna. She's at the NIH. She works with Dr. Fauci and Dr. Graham, and she has been instrumental in bringing their vaccine data and project to fruition as well. So two very important women. Hopefully this will encourage more young women and girls to go into science and continue our quest for new information on all different levels. This is my own animation version of what an mRNA vaccine is. It's very simplistic, but I hope it explains so that you understand exactly the technology. This is the SARS-CoV2 virus. These are the spike proteins that are on the surface. What the scientists have done is they take one of those spike proteins off, and here it is right here, and they basically take the recipe for that spike protein, the genomic recipe, the genes that are used for the virus to make this particular piece of protein, the spike protein. That's then wrapped in a little envelope. That's the piece that Dr. Kariko was able to conquer. And then that genetic information is put into a human cell. It's injected into the body. It goes into your cells, the cells that are circulating around. It never goes into your nucleus, which is where your genetic material is being kept. It just stays out here in the exterior of the nucleus called the cytoplasm, and it goes through a process through what's called the endoplasmic reticulum. And what that does, the recipe teaches the cell to make the spike protein. So now the spike protein's been made by this cell, and so that spike protein is then ejected out into the plasma, into the rest of your body, and along comes something called a B cell. A B cell is a very important part of the cellular immune system, and it makes something called neutralizing antibodies, which are proteins that are specific for a particular protein. In this case, it recognizes the spike protein and says, wait a minute here, that's not me, that's something foreign, so I'm going to make an antibody against that spike protein. So then along comes the real deal. You're exposed to SARS-CoV2. So now the body knows how to make proteins against the spike protein. So the B cell comes along and goes, uh-oh, there's spike protein in the vicinity, which is on the surface of the SARS-CoV2 virus. I'm going to make antibody. The antibody goes over, it attaches itself to the spike protein, and it kills the virus. And that's how mRNA vaccines work. What are their advantages? Well, they're very safe. There's no risk of infection or insertional mutagenesis. That means they don't seem to cause cancer. They don't get into your own genetic material. They're kind of vacuumed up by normal cellular processes, so that it gets rid of there's no extra material circulating around that could cause problems. You can adjust them both in the strength of the immunity that they elicit and also the targets. So for example, if we're starting to see more variants or mutations with SARS-CoV-2, we can have the ability to dial up these vaccines pretty easily. They're efficient in taking the information into the cell through these little carrier molecules that Dr. Kariko was so instrumental in discovering. They also can be repeatedly administered so that there's no material that's accumulating that's causing effects. So if we have to re-administer things, give people a booster shot, or give another variation if the virus is mutated, that can be done fairly easily. And they're easy to make, inexpensive, and you can scale up production, so you can make lots of it pretty quickly. The disadvantages, they've never really been tested during a pandemic. That's what we're going to do now. There may be some unforeseen issues down the road that we don't know about. There could be, they haven't been very well tested in humans. We have some pretty big trials. 40,000 people in each of those trials is pretty big, but it's not 40 million people or 400 million people. So as you increase the numbers of people receiving these vaccines, you're going to see some things that you didn't see perhaps when the studies were smaller. Are we seeing that now? Maybe, but nothing terribly deleterious or dangerous. And so it's just information that we have to follow. mRNA vaccines have been developed for other pathogens, including HIV, Ebola, and Zika. They were successful for the Zika vaccine, but not necessarily for HIV. That's a whole different problem with HIV, and it's been a challenge to develop a vaccine. And perhaps more importantly, we don't know how long people can retain immunity with these vaccines. But we have two of them. The first is the Pfizer vaccine, which both of these, as also the AstraZeneca vaccine, which is an adenovirus vaccine, all three of those vaccines require 2 shots, what we call a prime and then a booster. For the Pfizer mRNA vaccine, the prime is given first, and then 3 weeks later, the booster is given. For the Moderna vaccine, the prime is given, and then 4 weeks later, the boost is given. I think the AstraZeneca vaccine is also 4 weeks apart, but I don't know for sure. The Pfizer vaccine, I'm going to present the data for that, but it's almost identical to that of Moderna. There were about 40,000 people recruited, about 37,000 of those individuals were enrolled in the study. And it was a pretty wide range, 16 to about 55, a little few people in the older 60 and 70 category, about 43% males and 53% females. It was fairly diverse ethnically. But again, it's always hard to recruit a large, very reflective population when you're doing studies like this. But it was pretty representative of the population where the study was being done, which is mostly in the Southeast, but throughout the country and in Europe as well. What you can see here, this is the final result, which is really astounding. So as the independent review board was monitoring the data, what they saw was all of a sudden, we started seeing cases of COVID going up and up and up and up and up and up and up. And then some individuals, it wasn't going up at all. And what that showed is the individuals that were getting COVID were all the people that were not vaccinated. They're the ones that had received the placebo. Here's the group of individuals who received the vaccine. And you can see that there were a couple of, about 4 or 5 cases, but out of 197, that was a very, very significant result. In addition, they went back and looked at the level of neutralizing antibodies, the first thing that you respond to when in your immune system. And they could see that over time after vaccination, there were very high levels of neutralizing antibodies in individuals who had received the Pfizer vaccine, comparable to those, if not better, frankly, than those individuals who had survived a natural infection with COVID-19, with SARS-CoV2. This is my representation of the antibody responses, so it's a little bit easier to see. You get your first vaccine here on day 1, and then 8 days later, you do have a pretty good response. You've got maybe 40 to 60% of your antibody responses already beginning to develop in that first week. Just before you get your second shot with the Pfizer vaccine, remember that's 3 weeks apart on day 21, you have about 65 to 80%. Some people think that may be enough to protect the population, that maybe we really only need 1 shot rather than 2. The British are betting on that right now because they're having such a terrible pandemic. They're trying to utilize get as many people vaccinated as possible. But in the United States, we are recommending that you get the second vaccine because that really bumps your response way up into the 90s and higher, which is a very, very good antibody response. Indeed, you can see that that's higher than even natural infection. And for that reason, we recommend that individuals who've recovered from COVID-19 still get vaccinated about 90 days after their disease because the vaccines are more likely to produce immunity than natural infection. Pfizer also looked at the cellular responses. Remember, the T cells that come in, they stimulate the B cells, which make the antibodies, so that's very, very important. This is where memory for the immune response is kept, and so this is a very important part of vaccine analysis, and indeed, there were very good cellular responses with the Pfizer and the Moderna vaccine, enough to certainly probably have a vaccine at least be good for about a year or so, but we don't know the true length of time. Side effects were relatively minor. Many individuals experienced a sore arm. I have had my vaccines. I had a sore arm both times. You can get a low-grade fever. That's very common. Some people get a little bit higher fever, 102, 103, and feel rather fluey. This tends to occur after the second shot because, of course, that's your immune system responding. Muscle pain, joint pain, and swollen lymph nodes are fairly common. Sometimes redness around the injection site, which may take a few days to go away. Those are all very common and really relatively minor side effects. There have been a few cases of severe allergic responses, or what we call anaphylaxis. It does appear to be very rare, and we still think that you can vaccinate those individuals, though you need to take precautions. It should be discussed with your doctor if you have a history of anaphylaxis. What we do recommend is when you get vaccinated, the clinicians will watch you for 15 minutes. You're required to remain in the vaccine site for 15 minutes. If you have a history of anaphylaxis, you're watched for 45 minutes. because that during that time is the most likely time that you'll have that kind of reaction. We are beginning to see some things that are happening in individuals. There may be some rare events that happen as you begin to vaccinate more and more people. You begin to see things that perhaps weren't picked up with the first trial, but those appear to be very rare. They may include some hematologic issues that we're looking into as we move forward. But for the most part, these vaccines appear to be very, very safe. Because the vaccine production is still ramping up, there has to be a tier system in terms of who gets the vaccine first. Healthcare workers have been designated first, along with residents in the skilled nursing facilities and their care workers. These are very either individuals who are likely on the front lines, who are likely confronting a lot of coronavirus infection, or they suffer the consequences very severely with coronavirus infection. But as we are seeing over the next few weeks, we're beginning to ramp up vaccinating first responders, the police, paramedics, all people in healthcare personnel, the 75 plus individuals, people who have comorbidities, people who are immunocompromised. Eventually we'll get to teachers and college students, and then everyone else. And I think that it is the plan of this administration to probably try to get as many people vaccinated within the next 2 or 3 months as possible. There is some urgency in getting as many people vaccinated as possible, because the longer this virus continues to be out there circulating, the more likely it is to develop mutations that can cause either worse disease or cause some lack of efficacy of the vaccines. Special considerations would be pregnant women. About 9 individuals during the Pfizer trial became pregnant. They gave birth to normal children. Of course, pregnant women are generally not part of phase 3 trials initially, because it's a very delicate situation. But the American College of Obstetrics and Gynecology do recommend not withholding the vaccine in women who may be at high risk for acquiring COVID-19. We have had some women who've become infected with COVID-19 who are pregnant. There have sadly also been some women who've died 2 or 3 days after giving birth to what appear to be normal babies. Really a very sad situation. So probably in the long run, these vaccines are safe and should be given to pregnant women. The American College of Obstetrics and Gynecology thinks so, but we don't have a lot of data to support that. Immunocompromised people are probably fairly safe. People who are on cancer chemotherapy, on immunosuppressive medications, who have HIV. We know that these are not live virus vaccines. They're an inert genetic material. They shouldn't cause any kind of response in a person whose immune system is compromised. And we do know that these individuals are at high risk for a bad outcome should they become infected with SARS-CoV-2 and get COVID-19. Children, we still don't know, have all the data. There's a study being done right now with about 2,000 children, but it does appear to be safe in children. It's likely not to be any kind of significant side effects. It just hasn't been studied very well. Children certainly can get COVID-19. Sometimes they can die from it. They are also, of course, an important super spreader of the virus. And so it is important to consider vaccinating children as soon as we have some information that it really is safe, which I think most of us believe it will be. People with underlying neurologic disorders such as Guillain-Barré, multiple sclerosis, ALS, could be a little bit more problematic. We don't know if these vaccines could elicit a second inflammatory or response that might exacerbate some of these conditions. It's not very clear. But we also know that individuals with these diseases, again, are at high risk for a bad outcome if they get COVID-19. So you're always weighing the risks and benefits of the vaccine versus the disease. I mentioned anaphylaxis. We do think that people should be vaccinated, but you have to watch them very carefully, and that should be something that you discuss with your physician. If you've had a stroke or other type of event like that, then this vaccine is indeed safe, and we know that COVID-19 can cause, the disease can cause stroke. Well, how long will the vaccine last? We don't know. That's a question that's going to be very important going forward. We do think that you probably will retain antibodies at least for a year or so. There are some good data looking at individuals who've had the disease, and that appears to be the case. We're up to about a little bit short of a year in terms of Americans that have had COVID-19. But we do think the vaccine will probably work at least for a year. It might require a booster. We're not sure. That's data that's going to be collected over the next several months. We hope it lasts longer. Perhaps it may be a lifelong vaccine, but we need it to last long enough to control the spread of the disease. And once that happens, then the virus really doesn't have anywhere to go, and it will eventually die out and dissipate. The news media has been focused a lot on these mutations and variants, and they are of some concern. This is a natural phenomenon with all kinds of viruses. We know it happens very quickly with influenza virus, but it certainly can happen with coronavirus as well. They naturally mutate, and they collect genetic mutations that may or may not be helpful to the virus. If it's helpful to the virus, then that often becomes the dominant virus. We're certainly seeing that in England with the B117 virus that also probably is circulating in the United States. The British are better at monitoring and surveilling the presence of coronavirus mutations than we are, but we are ramping up the CDC SARS-CoV2 strain surveillance service, which will try to continue to monitor the emergence of mutations. There are a few other mutations that are being noted right now, one from South Africa, one in Brazil, actually two in Brazil now, and then one recently found in Santa Clara County that may also be circulating in LA County, the L452 R variant. All of these appear to be more infectious. They're kind of stickier, if you will. The spike protein is able to attach a little bit more robustly to the human cell, so that makes them a little more infectious. We don't think that they cause more severe disease. They do appear to be covered by the current vaccine, although it's not been extensively studied. We certainly know with B117 that's probably certainly captured by this vaccine. We'll have to be vigilant to make sure that there aren't strains that end up requiring vaccine adjustment. But that is the beauty of an mRNA vaccine, is that you can adjust it pretty easily. Of course, to control this pandemic, we're going to have to control the disease around the world, and that's going to be a Herculean task. Many developing countries don't have the resources or the monetary and or the medical resources to do large-scale vaccination. It's going to be on the shoulders of developed countries such as the United States, those in Europe and China and Russia, to really help assist smaller countries that are going to need this. Because if you have a country where coronavirus continues to circulate in spite of the fact that everybody else in your country has been vaccinated, that will continue to perpetuate the pandemic. So it's going to be a really important part of getting this pandemic under control, a tool that was not available during the 1918 pandemic. We do need to make sure that all communities, even in our own country, are served by this, that the vaccine information is available to them in ways that they can understand. They shouldn't be afraid of the vaccine. We want people to get vaccinated for this disease and many other diseases. Vaccines save lives. They very, very rarely take lives, but they often save lives. And so it's very important that we try to get this information out. Our project in Tanzania is certainly focused on other diseases besides COVID, but we hope as we move forward over the next few months and years that we will be able to help them address their own pandemic in a way that will provide good, sound science information, scientific information, and both clinical information and perhaps hopefully vaccines as well. So it behooves all of us to be good citizens of the world and spread information, look at the facts, look at the science, understand what the process is, ask good questions so that we can move forward and finally get back to some sense of a normal life. Vaccines won't be the entire story here. We are going to continue to have to wear masks. We know that individuals that are vaccinated may still be able to spread the disease. Hopefully that data will be coming in the next few months and will tell us that that's not the case, but we don't know for sure. We certainly need to practice social distancing and good hand washing. Those are things that I think society will take forward at least for the next couple of years as we all sort of try to recover from this very traumatic event that has happened to us. But hopefully it will bring us all closer together and realize that the legacy of those that we've lost to COVID-19 will be what we have learned through the truth and science and equity, so that the next time, and there will be a next time, we can and must do better. And with that, I'm going to close. I think that this is a journey we will all take together, and we will get to the other side, but it is going to take patience and perseverance and strength, equity, good science, and good governance. Thank you very much.